Now, the extra slides that I had sent out were just from the NIH or National Genomic Institute website. The current head, physician, researcher named Eric Green, he has done that after 20 years. So those are his slides, that you saw. And that goes through the, the thing that they went through for the Human Genome Project. It summarizes in like 20 slides, so if you're interested, look through that, it tells you about, so a lot of the political things that went back. So I think we stopped somewhere along. We talked about the classical genetics, the Mendelian genetics, and then we went into the rise of molecular medicine from 53 to about the rise of Genentech and the GMOs and the protests that took place mostly in Europe, and we didn't get too much into genomics per se. So we'll delve more into it and how it has made a huge difference. Because obviously, so we're all interested in heredity, basically, type of thing. And this is, this was reading the genetic sequence, that was not so easy, but was done with the Sanger method. And the other method that revolutionized, we'll talk about it in this class, okay. And this is, so we're going to be talking from heredity, from gene to genome, and how it affects not just reading the pure scientific point, but the consequences of the changes that take place and importance in health and food, that as we talked about, type of thing. And we already talked a couple of these factors that influence heredity. We already addressed that: mutations, the varieties of mutations, the so-called harmless and the harmful ones, and the mode of transmission we have discussed already. This we have not discussed. Symbiosis, I already alluded to about Lynn Margulis, who proposed the concept. And so those are some of the concepts that we have already gone through. Hybridization, okay, which is mostly done in plants, but it also occurs in, mammals and eukaryotes, and that we'll just address too when we talk about the evolution, type thing. So there are varieties of other subjects that I've done in other classes. I'm not going to be able to cover at length. For example, I think the genes and aging I won't be able to cover at length because everybody has huge interest in telomeres, and that got the 2009 Nobel Prize. So, but we won't dwell too much into it. At the same time, we did a class on history of sex, or the reproductive genetics type of thing. Some of the words that we won't have that much time to. I may make a reference to it here and there. Okay, so and obviously, the aging, also we talked about in the other classes. So, okay, but so this is just to show that I'm just not just a science nerd, just to show that I do have a little bit of culture. I threw in a poem by William Blake. I have no idea who that was. Well, actually I do, but I think some of it. This is not his more well-known poem, but it tells you the things about how the similarities between the smallest life form and human. At that time when he was alive, the humans were considered at the top of the chain, the Linnaeus classification, and the concept was that the animals and the other species did not have souls, so that led to a lot of vivisection. And at the same time, we can just see with the mutation, can go right. This was one of those movies, that the starring called Fly, which came out, I think, in the 80s or something, so as to how the two genes get mixture. And so the two elucidation of the fly as to how it plays a role in the evolution when everything goes as nature's plan. And at the same time, when and this will figure more when we're talking about the gene editing in the next class. Okay, it comes at us to how some of the earlier efforts had backfired onto us. So this just kind of lays the ground. Okay, and I think we won't do the analysis of the poetry, but I think that you could look up the Spark notes if you feel like. I had promised last time that that the intracellular signaling networks and pathways are complicated. So I just added in this side, so this is just shows us to this makes the London, a metro map look, more easy to figure out. So this is just one example, and this is, and this is one of the things that I quoted last time. Biology is messy, but you think biology is messy? Biochemistry is even more messy, and that's why a lot of pre-med students drop out. So with apologies to the two biochemists in the class, so this is, now I don't expect people to, but this is, again, just shows as to how all the end of stuff that we are interested in is the nucleotide and protein metabolism. And this has to do with our food, and diet. So just so as to how everything goes down basically into the oxidative phosphorylation. And this also takes into account the energy that we talked about. We kind of glossed over it when we were talking about, the basic DNA and the genetic code. Okay, but this gives you some insight as to how complicated networks can be. Okay, and again, as to how and the prototype animal was the fruit fly, that started with Thomas Morgan, and it has given us a lot of insight. But there were a group of scientists, I think they're Germans, I think, yeah, and they were interested as to how the code is basically same, for most of the eukaryotes, but how the different organs, how does the part, what genes decide as to this part should be head and this part should be thorax and this part should be abdomen. So they did a lot of the, I'm sorry, my mouse clicked bad, okay. Okay, they did the fundamental works with looking at thousands of mutation experiments and determine as to how the patterns of segmentation of fruit fly was occurring, and looking at it meticulously at each mutant under microscope, and we were able to work out which a group of genes were involved in different processes during development based on their phenotypes. I'm sorry, that is expression that took place, and the normal segmental pattern as to how that whole orchestra was coordinated in a fine symphony, so to speak. And again, as we saw that if you discover a gene, you're allowed to name. And so this is when they came out with the names such as sonic hedgehog, okay, and being gherkin, which is German for cucumber, and Krupfel, Kruppel, which is cripple. So the varieties of names were identified, and they were for this classical work on analysis as to how the body plan is formed. They were the Nobel recipient, and this was and the work was done, back in the mid 70s. Okay, and okay, excuse me, right. And this is again, some of you who taken maybe, I don't think this was taught when, at least when I was doing basic biology, but this is how the gene plans as the homeobox plan as to how nature is very conservative, that what has worked, it is preserved, as to so that's, I think, William Blake, even though he was not a geneticist, he had a very point that we have a lot of similarities with the basic, simple, organisms such as fruit fly, is to see how some of the genes have been conserved, through the evolutionary, and through comparative genomic analysis. This has been proven time and time again. It has been the big, and obviously this has advanced a lot more far, but in the earlier days, it was a very complicated task because they had to meticulously isolate the gene and observe the phenotype under microscope. And these are the various parts, that how they worked in pairs or in a cluster of genes that worked in harmony and created the antenna and some of the mutations that we done as to how part a leg can grow on the thorax or eye can go in the body part. So some of the stuff that can be done also. So that just proved as to how the body plan was laid out and was conserved from not just from fruit fly, but also in mouse embryo. And you can see these are nicely color coded and into the adult human as to how the essential parts are conserved. Okay, so that's the point, So, and this is controlled not by, but this is controlled by varieties of proteins because, as you can see, genes by themselves, are not the real work is done by proteins, and there are a variety of protein gradient that control the expression of these genes. Okay, and again, when you can say about the numbers, that now we know that a lot of scientists were disappointed that human genome had only like 22,000 genes, but it may be a sobering fact that we share almost 3,000 genes with fruit fly, almost 2,000 with a nematode, which is the work again another prototype for work called C. elegans, and so these and all three of them share as many as 1,500 Hox genes, Okay, and then again, it gets very complicated. The varieties of transcription factors, which are the varieties of proteins that control the expression. So that's why we don't grow an antenna, but we grow limbs. And this is interesting that quite a few who may remember the thalidomide disaster, which was a drug that was given for preventing nausea in early pregnancy, and fortunately, that was never FDA, the physician who was in charge, she stood her ground and did not approve it in the United States, but the Europeans and South Americans had approved and caused the abnormality of the kids who were born with deformities of phocomelia, so like flipper arms and legs, basically. And after 60 years, that has been isolated that there is a gene called SALL4. The name is academic, but that was the responsible. So that transcription factor and the gene was mutated or affected by thalidomide. And so afterwards, in a molecular basis of thalidomide disaster has been identified. And similar factors maybe occur in puberty also. Because, for example, like in a butterfly, metamorphosis takes place, and some of the expression of the genes that takes place at a puberty level and causes the release of male and female hormones. Okay. So one can say that puberty is a sort of metamorphosis that takes place for humans. Okay. And again, it's like if you ask a layperson in the street, their genetics information from watching the NCIS and CSI shows would end at DNA, and I think Ollie had a nice class about the DNA lab for the local FBI chapter. I think I didn't take the class, but I think my wife did, and they showed how the analysis was done, And this was discovered by an English geneticist, not sure, but named Alec Jeffreys back in '84, and he described. He was in 1984 in Leicester. He was looking at film images of a DNA experiment that was done, and it showed similarities and differences in different members of one of his associates' family, and he realized that these can be used for identification. Okay, and okay, and so this was the start of the DNA profiling technology. Okay. Varieties of tandem repeat segments, because again, as we said that the eukaryotes, signaling is not exactly like, for example, the word genome is not transcribed as genome, but it's transcribed as G dot 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 NO dot 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 NOM. So non-tandem repeats, and so these segments, there are so many varieties that takes place, and so mathematically, it was unlikely that they would be similar in two people. So initially they were comparing about 10 not tandem repeats, but now it has been raised to 13 to avoid any chance of real random chance error. And this was used to solve a famous murder in UK about a guy named Colin Pitchfork, and who was, one of the first person to be convicted based on this. And this was also used in immigration cases about how the UK authorities were denying a child who was born in Africa as to if they were not the real parents, so to speak, and the data was used. So this was the start of DNA profiling, which is the workhorse of criminal investigation. Okay, enough about that. Now, again, this is, I'm not going to show too many videos, but this was too far out to avoid. So
whether we could use this air DNA as a way to assess what species were present in a burrow or a cave when we could not easily see or capture them. Lots of scientists had speculated about this, but we could not find any published case where someone had actually tested for animal DNA in the air. Well, our paper describes really the first proof of this concept. We've experienced filtering eDNA from water, so we used a similar method, sucking air samples through a really fine filter and trying to trap air DNA. We sampled air from inside the artificial burrows of some naked mole rats, and then in the room where the burrows are housed. We then extracted the air DNA from these filters, and we sequenced a piece of DNA which would let us identify species from that sample. We compared this DNA against reference databases and identified the naked mole rats, but also humans who care for them. We found the naked mole rats exactly where we expected them to be in the burrows, but also in the room, which shows that the DNA is moving away from the source. We also found human DNA in all of our samples, and this was something we had not anticipated. At first, we considered this a contaminant, but what we realized is it opens up some interesting questions about how this could be used in archaeology or criminal forensics. The biggest challenge we anticipate for future applications of this methodology is the potential that the air DNA is going to be diluted. In a large space, it might be so diluted that we can't detect it efficiently. We'll need to suck lots of air through a filter to accomplish this, but in a smaller space, we think it might work really well. I think this is going to be really interesting to scientists who work on animals that are just hard to get to. Roosting in tree hollows, underground, small, hard to reach caves. It could let us monitor the species present without actually having to disturb them or get really close. However, the unexpected side effect of collecting human DNA is also really interesting, and we're now discussing what we can do with this sort of technology. Our study suggests we may have another tool we can use in our toolbox to measure biodiversity. We're desperately in need of ways to monitor species on a planetary scale. We think what we have done can be done in remote areas and in
Okay, so you get the idea. So this was, this is far out. So I mean, I just thought this was really interesting type thing. So, and again, the classical things about human nature is this is again, I want to reemphasize that this is not all, genetics, but again, the environment has influence also. And I just recently finished a book, which talked about the neuroscience of intelligence, and some of this interesting stuff. In our great books class, we read about inheritance of intelligence type of thing, and there are some, and this had the bad stigma during the early days of eugenics, so that subject is considered. A lot of people that it's a career-ending move if you're doing research on, but it may be making some comeback type of thing. And the earliest data that was done goes back to Galton, and he had started twin studies, but the more systematic approach was done with the more famous one is called the Minnesota twin studies of identical fraternal and twins who were raised separately, and they've separated at birth, and it gave the insight as to how, heredity may be playing a bigger role than you realize type of thing. And again, the guy who got the bad publicity back was, I should say, probably in the 80s or late 70s, a guy named Jensen, whose report was considered very, controversial. And still some of the newer work that is done by a psychologist who actually was, had enough flag returning that he's American, but has worked in UK now. He's doing work on intelligence, a guy named Plomkin, and he has done a lot of interesting work on genetics and intelligence. Okay, so so there are one can say that there are roles of other factors such as epigenetics that we'll talk about in a few minutes. That and some of the European countries, there were better data on larger studies and databases. And one can say that the genetics puts it together, while the other things, such as epigenetics and microbiome information, pulls it apart. So it takes an interplay of all three factors put together to make us who we are, so to speak. Okay, so and again, for other diseases also, twin studies have been a big boon for collecting data, such as, for mental illnesses such as schizophrenia. Okay, and when one of the identical pair suffers schizophrenia, the chances that the other twin will have a schizophrenic is about 1 in 2, and 50 to 60%. Okay, it's 1 in 6 for fraternal twins. So this is so there is a genetic tendency type of thing for a lot of these mental illnesses. And then we'll talk more about the other complicated data, such as polygenic score, as we get further into the talk. Okay, and this was interesting type of thing. I don't know if you can read this. There were identical twins until John joined the army and another joined the Peace Corps. Okay, so I thought that was interesting influence as to how the external conditions do influence our behavior. Okay, and this was if the biggest discovery in biological sciences was Watson and Crick, but this is right up there, type of thing. So this has changed how the genomic analysis is done, and this is a rather complicated slide, but this was, the start of as to how DNA analysis became, okay, because this was done with, denaturing the sample, as well as annealing, means heating and allowing it to cool slowly, and then adding varieties of primers, it means the small nucleotide elements which are attached. And after running through varieties of cycles, the amount of sample increases exponentially. And for example, running like 25 cycles, we have like a 30 million fold increase, okay, in the amount of sample that was collected. Okay, and there are varieties of enzymes, which are collected from the bacteria, which are a lot more stable. And this was the procedure that the idea came to a chemist working for Cetus Pharmaceuticals when he was driving on the Pacific Coast Highway, a guy who is a colorful personality named Cary Mullis, in April of '83. Okay, and he did subsequently the experiments on that, and that was the start of the PCR reaction, which is the workhorse for all DNA analysis. Okay, so and this was again then later on by adding varieties of dyes, this reaction was automated, and by adding variety, and that work was done at Caltech, and it was commercialized, and that's the big underlying mechanism for doing DNA analysis and the huge machines that are providing the genome analysis. Okay, all right, and again, so you already seen the slide for the Human Genome Project, and right around the same time in the '80s, people with the technology, it was ideas were brewing in the molecular biology and genetics field as to we should, aim high and try to sequence the human genome, and the varieties of as you might have seen in the slide that I had sent out, that the organization was done. Some people were in favor of it, others thought it was too technically challenging, and on the East Coast, varieties of meetings were held, and eventually, with the backing of the Department of Energy, because they were interested in the effect of radiation on DNA, and this was still during the Cold War era, so they threw their weight behind it, along with NIH. And finally, with the congressional hearing, which were, in which James Watson gave the evidence in favor, the funding was provided, and then the Human Genome Project was taken ground. Okay, and the timeline was extensive from 1990 to, it took almost 13 years basically. So, and this, you can just see this slide basically. So I've divided up in various areas. Let's see if I can get my magnifying lens to work. Oops. Oops. Oh, hit the wrong button. Apologies. Sorry. Let me go down to the. Okay. Yeah. Yeah. It's hard to get to that little magnifying lens. That. Oh, I suppose I could do it on my smaller screen. Okay. Yeah. That works better. The Human Genome Project was launched, and the centers were established. Okay. And it was going to take. Sorry about the phone call. Okay. And that it's going to take a lot of time and money to do that. And initially they started out slow, and by 1995, the first bacterial genome was sequenced. Okay. And the map was established with pilot project. And then again, so this was variety. And it was a multinational collaboration with the famous Sanger Center in UK, and various other places all across the US, including WashU, MIT, and Washington University. So there were several institutions involved in the project. Okay, and they started out with H. influenza, and later on moved on to C. elegans, which is a small nematode, later on to Drosophila, the fruit fly, and the various places that were involved in it. UC Santa Cruz, Department of Energy, NIH, and other countries. Japan was involved. And the PCR, as I mentioned earlier, was the workhorse. And the big people were Watson, and then later on, after he fell out with the head of the NIH, Bernadette Healy, Francis Collins, who was a more savvy administrator, took over, and he saw it through. And at the same time, you may or may not have heard of Craig Venter, who started out with humble beginnings as a medic in Vietnam, and later on got at UC San Diego, got a PhD, and was working with NIH. And he decided to try a different method, okay, of analyzing human genome, which is called the shotgun method. And along with that, he obtained financing, and his idea was to monetize the process, and that did not go well with some of the other people. And his method, as it was so-called quick and dirty, but was prone to some errors that could creep in. And some of the more traditionalist people who wanted to do a more systematic search were aghast at his ideas. And the two were initially in dramatic competition. And for a while, it looked like Craig Venter might succeed and be first. But then NIH, through its weight and more financing, and eventually with the intervention of Bill Clinton and his team, a peace was called, and finally both of them, announced it simultaneously. So it's a very intriguing story of the how the Human Genome Project came to be. And again, some of the more dramatic headlines that the New York Times gave it a full 8-column headline, and along, some of the British newspapers. I mean, was almost over the top, sort of opening the book of life, and a discovery that will touch every person on the planet. I think we're waiting for it to touch us. Yet one can safely say that it wasn't as straightforward as we thought. And it hasn't affected each and everyone's life. Okay, I think anybody has any questions or comments? I think, oh, I'm sorry, I should have been seeing this. Okay. Oh, and oh yeah, this was Peter's comment about the obituary that we were chatting about earlier. Okay. All right, thank you. Yeah, I'm sorry, my mistake. Okay. And so this is again, and and then there was a race as to how many genes a human genome will have. The people were taking some wide guesses from anywhere from a few thousand to 100,000, and eventually only very few people came together. So type of thing, and it was a somewhat of a letdown that we were not didn't have a lot more genes than humble fruit fly, and some of the plants have even more genes. But as to how how they are put together, how they function in harmony, that's the important part that we'll get into as we go along. Okay, and all right. And some of the venture capitalists, wanted to finance this type of thing, and that's how the name Craig Venter's company was named Celera, and which was kind of a play on the words on accelerate, on speed type of thing. So that was the name of his company. Okay, and with some of the very early pioneering computing guys actually did a lot of the backbreaking work. Wellcome Trust programmer named Jim Dent, he actually put together 100 personal computers in the days of Windows 95 to finish the draft, a rough draft, on June 22nd of the Human Genome Project. So it took, a huge amount of almost, sort of moon landing type of effort to finish the Human Genome Project. So, okay, but, and this was as to whose genome they should take. So this was a very humble and advertisement that was taking place, wanted 20 volunteers. And eventually, I think there is a name available, but I didn't write it down. So, sorry, my mistake as to whose was initially. And then Jim Watson was one of the genome that was analyzed later on by one company also. So he had he revised his book, the DNA, which he had written back in the 60s, which he's revised it, about DNA from the beginning, and in collaboration. And he gives a very interesting story. And in the second version of his book, he gives due credit to Rosalind Franklin, which he actually hadn't said much in his first book. It's an interesting read if you are interested, you can read. And this was an interesting as to how it was printed out in the old style book. And this is what it would take, is to print the whole human genome. So you can see how many books it would fill. Okay. And this was, I'm not sure as to what newspaper this was advertised. Okay. And then later on, there were varieties of project, which was ENCODE project, which was called Encyclopedia of DNA, that was started, around the same time, as to see how much of DNA function took place. Okay, and all right, now getting to how big is the genome? Okay, obviously, we're 6 billion letters, so 3 billion bases. Okay, but to the disappointment of many geneticists and scientists who work, there were only about 20,000 protein coding genes. Okay, and there are varieties of other stuff that was initially thought or labeled as quote unquote junk, or DNA was not doing anything, but we're still finally learning as to what all these fragments are doing, as to how they are controlling the expression of the protein coding genes. And so it's finally, being sorted out, and we are learning the significance of the non-coding elements. Along with, there is something called pseudogenes. Okay, and that a good example that would be that in the human in the primate evolution initially, the classic example would be we were able to synthesize vitamin C, but then somewhere along the line we have lost the ability to synthesize vitamin C. Okay, and that the deficit was very apparent with the disease scurvy that manifested on long sea voyages. And that may have been due to, on an evolutionary basis, with the availability of easy availability of fruits, as humans, as primates got down from the trees. And that's why that defect was not easily missed and was carried on. Otherwise, any harmful mutation would generally be eliminated. But since it did not affect, the well-being of the species, that gene was not eliminated. Okay. Now, again, so this rather complicated slide. I put it in because this was the photo of Jim Watson and Eric Lander, who was fired as the science advisor, in the current administration, and Francis Collins. I don't know who the next person is. And this is an interesting slide, which is rather very complicated, but I put that in just for the sobering thing as to, number one is the density. So varieties of band as to how the various genes are located. Number two is the segmental duplication as to how gene duplication has played a lot more role in forming creating the human genome. And then number three, that the red fragments are the same elements that you have in common with a simple mouse, another mammal. And last but not the least, just to show number four, that you have things in common with the poison of fugu fish, which are in the non-coding region. So you can see how the elements which goes back in the evolution, biological evolution, has been conserved in the chromosomes. And this is still, the sizes of this are different. And let's see what else I have. Okay. Yeah, and we've already talked about the struggle that went on for the thing, so I won't repeat myself, so. Oops. Yep, okay. I had another slide in here, so. Okay, yeah, all right, I will skip that part because I think it does not play nicely. I thought I had one picture underneath it, but it does not slide. Okay, I'll try to fix that and send it in my batch of slides when I send out. It had pictures of other chromosomes as to how some were small and some were this thing. So, oops. No, I can't slide it horizontally. And again, genetically, these are classified as the P arm and Q arm. So if you look at the analysis of the location, as like, for example, the address of a particular gene on the chromosome, it would be identified as P, Q, and number 22 as to which segment. That's how the gene is identified exactly. So if you look in the database, okay, all right. The other part that is people had glossed over, and because simply we're not aware that how much viruses have played a role. Okay, and I think we talked about it when we talked the genetics of sex in that class, if somebody may recall, that human placenta, that some of the 8% of our genome is actually endogenous retroviruses that somewhere along the line that made peace with the human genome and became part of the human genome. That may come as a shock to some of us, but actually that huge part of that has similarity with endogenous viruses. The so-called exogenous retroviruses are the ones that cause disease, but the ones that are there have made peace with the human genome and have become part. And the classic example so one can see that how the microbiota, which includes eukaryotes, bacteria, archaea, which we had mentioned, and when archaea became part of a bacteria, that led to the formation of eukaryotes. But underneath that, there was a huge large circle that we did not discuss, that initially people were not paying that much attention to, is that the viruses, and underneath that bacteriophages. We discussed what the difference between phage and virus was last time. Okay, and so this have become, there is a substantial overlap, and the varieties of conditions where either they're parasitic, it's cause disease, and have lived together in mutualism. Okay, that thing. So there is, and the big element that has taken place is the endogenous virus has become part of the placenta and forms a protein called synectin. Okay, and that's one way the fetus may have a different blood group. At the same time, a fetus is able to take nutrition from the mother because the blood does not actually mix directly. So the nutritional needs, including oxygen and other nutrients, are provided by this particular protein that forms a barrier on the placenta, and that gives rise to the live birth that takes place in mammals. And this may have occurred as far back as 130 million years ago. Okay, so this was essentially a viral protein that morphed and evolved into this particular gene and protein, and that gives the growing fetus chance to fuse the cells into the wall. Okay, so that's an evolutionary symbiosis that has taken place and played a big role. Okay, and this was the big announcement that was done simultaneously in US and UK by Tony Blair. Okay, and about the Human Genome Project. The date was well marked. You can see it for yourself. And there's a 40-minute YouTube speech, type thing. So, and again, this, as somebody who's not a big religious believer, this was interesting to me. So the universe today, we are learning the language in which God created life. Okay, I think so that was, I had to say. So this was that was the one of the quote from Bill Clinton's speech at that time. So, and he thanks Jim Watson and the other scientists, and so this was again. So this was an announcement, but again, it was a lesser known secret that the project was far from finished. They did not have a lot of areas which were still, in the dark, so to speak. The in between part called the centromere and the part of the Y chromosome, they were not, and because the technology wasn't there, so and finally, so it was far from finished. But again, they had enough information to make the big announcement. Okay, and so and the earlier rough draft was finally finished in sometime in 2004. And again, these were very limited samples. There was lack of diversity that the others human races were not analyzed. And again, so this is, but at least that was a start. There were a lot of gaps that were left in between. Okay, Darwinian evolution, human ingenuity, which actually did a lot in terms of completing. So I don't know if God had much to do with it. Okay. So again, so we've come a long way from 2001. Okay, that finally it was actually this year that something called T2T project was finished. That is telomere to telomere. As some of you may know, the telomeres are the end pieces of human chromosome. Okay, and that has a lot to do with aging. That's another story by itself. Which I didn't put too many slides on it because we just don't have that much time to dwell into it. But finally, it was just this year that it was basically complete from something called the T2T project. So, but the work was going on with better sophisticated technology that took from 2001 until this year. Okay. And so it has created a huge database, and the big workhorse company for that is a company called Illumina, and the genomic technology and the huge databases that are created, that have helped finish the project. Okay, I'm just checking the notes. Yes, I think John's comment was, thank you, John. Yes, so I was making a comment about Bill Clinton. Okay. And the best thing that was done is that a lot of the genome sequences that are done is people are expected to upload, and it becomes an open source data bank. Okay, and so that has made a huge difference in terms of advancing the scientific knowledge, that if you have the enough technological or so-called bioinformatics wherewithal, you could upload your genome and compare it with the reference human genome, okay, and to see what the changes are. Obviously, it takes some reasonable background in bioinformatics, but the data is available in public domain. Okay, and these are some of the, this, we won't get into the technology of it too much, but this just gives you an idea as to how the sample is extracted, and there are with the varieties of things, the DNA amount is amplified, and varieties of libraries are prepared, and with a lot of automation, the sequencing is done. Okay, and you can see how the sequences are laid out in long stretches of alphabet soup. Fortunately, it's only 4 letters, and then at the same time they're uploaded and compared with the reference database. Okay, so this is the next generation sequencing principle. That thing. I won't show the slides of bioinformatics, but some of you may be aware, but there are a massive online learning free classes. One of my favorite sites is called Coursera, Coursera. You need a free account with that. And just this weekend, I just went through a slide on precision medicine that was done by University of Geneva. Okay, and the classes you can start on at random. You can start just this afternoon, so to speak, if you're interested. And only you pay if you want a certificate, but otherwise they're pretty congenial. And if you just want to learn, you can just look at the slides and videos. And they have some interesting slides, and after break time, I'll share some of the slides with you. So, okay, so this is how the next generation sequencing is done. Okay, all right. Now I just thought that this was an interesting thing, that the genome. Now this is basically it's an abstract concept where we talk about the various databases, but this was an interesting slide that just came across recently.
Researchers have recorded the genome in 4D, tracking the location of DNA strands in 3 dimensions over time. Our chromosomes don't always look like neat little X's. A lot of the time, our genetic material more closely resembles a pile of spaghetti. Even in the spaghetti state, genes move further and closer with purpose, forming loops of DNA. Many loops of DNA in the sprawling genome are bound by cohesin molecules. The cohesin acts as an extruder, pulling 2 areas of DNA together and extruding out the loop from the material in between. By removing and then adding this protein, researchers made specific DNA loops disappear and then reappear. But cohesin really only affects looping that brings genes on the same chromosome into contact. A second, still undefined mechanism seems to bring genes from different chromosomes together. The shifting landscape of loops helps cells quickly change which genes are active. Understanding this dance could help researchers better understand and treat genetic disease.
Okay. when I when I saw that, it reminded me of the physics things about string theory. Said, okay, oh, but this really looks like string theory in a microscopic level, or at a bigger level, so to speak. These are a lot bigger than what strings are supposed to be, but it just kind of a deja vu is that all over again, so to speak. And again, this is for more systematic data as to the common features. That basically that's a code which is something called the open reading frame. And we already talked about the introns and the exons. The exons are the ones that actually do the coding, and the segments that were, this went back to about the early 70s when the nomenclature was devised. And then I had already, if you recall, we'd shown the slide about the promoter, enhancer, silencer, so this DNA structure was more complicated in eukaryotes than in bacteria, which has a nice little simple circle, and everything is easily lined up in A, C, and G. But there are varieties of so-called non-coding segments. Okay, so this is and the varieties of enzymes, and so these are the numbers for that are much more precise. There are like 21,000 protein coding genes, and there a lot of these are just alphabet soup right now, but okay, all of these have various functions type of thing. So these are the small nuclear and nucleolar RNAs, which are abundant, play an important role in something called splicing of the introns, means the segments that don't make sense are cut out. Okay, and then the other one going down the list called microRNA that play a role in gene expression. Okay, so as to when the orchestra, part of the orchestra, should start playing, that's where they may play the big role. Okay, link RNAs, the long intergenic non-coding RNA. Okay, and this was, remember when we talked about during fertilization between male and female, the X chromosome is silenced. That's what determines the sex of the whether you're going to be whether the zygote is going to be male or female. That is part of the long intergenic non-coding RNA. So that was already known, and we'll talk a little bit more about histones as we go about. And so this is how these are the large segments of RNA that are playing a lot more role than we realized in the beginning. And that is still a lot of the work is still being done as to people are with the RNA analysis that is done. People are figuring out the roles of these non-coding segments that were initially sort of terra incognita, that were there a long time ago. But there's still a lot of work is being done on as to there were no new proteins that were discovered, but there are a wealth of non-coding RNA segments, that had a role in expression of the gene. And as I give you the example, it tells the particular cell to become liver cell or brain cell, as the case may be. Okay, let me just check at the chat. Okay, yeah, now we're good. Okay, so that's, so it's a lot more complicated than you realize. Now, genomics is everywhere. A couple of days ago, I was watching Jeopardy, and this was one of the clues. Okay, 50% of our genes are regulated by this rhythm, from Latin for about and day. And the answer is, anybody wants to say it?
Circadian.
Yep, absolutely. Yeah, that was the clue. So, okay, yeah. So this was the clue type thing, and the winner did get it right. So, okay, so this is, and actually, I had to look up as whether it was still 50% or more. But the most recent data is there is a sleep and circadian rhythm researcher at UC San Diego. His name is Sachin Panda. Okay, and his data was that as much as 80% of protein coding genes have a rhythmic effect. So that, but at least the whole thing is that it was so it's a very intricately influenced by the day and night rhythm. So that's an interesting fact as to how how the sunlight and makes a difference. And that emphasized the importance of sleep. Okay. Okay, and this was one of the codes from that, so. Okay, and so this is the sequence basically. So I think, this is like 3,000 base pairs. So this is a slide taken from Dr. Eric Green's slides. Okay, this is just, a very small percent of genome basically. Okay, and now, then these are the, you can say these are the protein coding sequences, the greens, so, okay, and then you can compare it with other species, which are the red, which are the, conserved in other species. So you can see that how the certain elements that are protein coding in human genome that are conserved in other animals as well. Okay, so those are the ones that are preserved, across varieties of many phyla. Okay, and this has, because they are very important in health and significance. So that's one way as to how nature has used the areas, that wasn't discarded and rediscovered, so to speak. So this was a good example as to how comparative genomic sequences, which was done with, primarily with mapping the RNA in varieties of phyla, as many as, not just with humans, but with other mammals were identified, and the comparative sequences that was done, that, and along with that, and taking advantage of computational biology in individual cell lines, we were able to discover the similarities and differences that have occurred over the evolutionary tree. And this is a complicated slide, but it just gives you the idea about how comparative genomics and huge data science has helped us understand our evolution from the simplest organism to the present one. Okay, and I think we're coming up to almost 2 o'clock. So before we get into the RNA, we should take a 5-minute break, perhaps, and then meet in about 5 minutes. I will pause the recording and then we can start in another 5 minutes. Resume recording. All right. Okay, I will minimize all of you first. Okay, all right. So Kevin is back, so we can start now. Okay, all right. Now, we've already talked about the DNA classification, but I think my slide was a bit out of sequence. But that's an interesting story about it, so that I just thought was worth sharing, Now, this is, John is not going to be happy with this, but DNA is just basically an encyclopedia in the reference section, You cannot check it out. It is closely guarded by the tough librarian, You can make copies of it, okay, but you don't know if you are just starting out as to what type to copy. So that's where the problem starts, So that's where the RNA is like the chef, I mean, I was listening to a talk a few days ago, and it had a nice analogy that the DNA is the huge reference book that you cannot check out, Well, and then finally, the copies are being thrown out for various recipes, and so the chef is designing this thing. And this started out with back in the 80s, or something there, that a bunch of botanists or plant geneticists were trying to make a purple petunia because they thought it might be more attractive to collectors and investors, and they can increase the funding, type of thing. And there was, and to edit gene in plant was relatively easy, which I may have mentioned it, but something called the gene gun, which is gold-plated particles are injected inside the plant, basically, and that's how you can introduce a new gene, And so they were trying that, and they put the gene for purple petunias inside, but to their chagrin, they ended up with white flowers, okay? And this was puzzling to them, and it took them almost, 15 to 20 years to finally figure it out, that this was the protective mechanism that the plants, the flowering plants had evolved in case the plant cells get attacked by viruses. Okay, so there were various enzymes. So one can say that these were like the cops. It said, okay, let's get rid of the bad guys. So that's how the gene for the purple pigs was totally eliminated. That's how they ended up with the white flower. Okay, and so this was one of the things, that the gene was not altered, but the expression was, inhibited. And so this was the discovery as to how the RNA was playing a bigger role than they had realized. And so that's why I put in the purple flower. Now we've already said, the main RNA that we have talked about, the messenger RNA and the ribosomal RNA, and then the huge encyclopedia that we have discovered, the so-called non-coding RNA, which can be divided into so-called housekeeping genes, which are the transfer RNA and ribosomal RNA, which is the basic central dogma pathway that we have gone over that many times: the messenger RNA, transfer RNA, and ribosomal RNA, which is where the proteins are synthesized. So this is the classical Watson-Crick, central dogma. But these are the other, varieties that we went over a little while ago. The long non-coding RNA and the orchestra that, and there's a lot of work on being done on piRNA that we'll get to in a few minutes, type of thing. Okay. All right. So, and again, this is Venki Ramakrishnan, who I mentioned earlier, and he was one of the Nobel laureates, who discovered the ribosomal structure of the 16S and 40S RNA. And it was shared with Israeli scientist named Ada Yoneth, which we talked about, that thing. And interestingly, how some of the names, this is called pi or PV, PIWI RNA. Okay, and the scientists, you can say, well, they do have a sense of humor. The PV RNA is named for P element induced wimpy testes. That's how the name came about. And the SN is small nuclear RNA, small interlacing RNA, nucleolar RNA, and microRNA. So these are the varieties of names of varieties of RNAs that have the function primarily in controlling the expression of other genes. Okay. And we know we had, again, not to miss the Carl Booth that we talked about, who discovered the third domain of RNA. So it's, okay. All right, so this is, okay, and this was the work that was done by, Venki Ramakrishnan. I learned about the ribosomal structure that was discovered, the large subunit of eukaryotes and prokaryotes that they had come up with, and then the varieties of shapes of the RNAs. So that it looks like a hairpin RNA, the transfer RNA, and these are the shapes. But this is probably a lot more biochemistry than you care to know. Okay. Now, in terms of technology, that this was, again, as to how varieties of technology of deciphering the sequence or DNA sequencing or RNA sequences. So as we already talked about, the PCR was this thing, but this was, again, the UK scientists must have a show for getting into in their pubs. And these are the two Cambridge scientists named David Klennerman and another guy whose name is even more complicated than mine, named Shankar Balasubramaniam. This were in the mid-1900s. So they used fluorescently labeled nucleotides to to observe the motion at a single molecular level, as the DNA to fix on the surface. And with variety of discussion, they came up with a method of parallel sequences of short segments, of DNA that were synthesized in the lab. So that was the new methodology that they came up with for DNA analysis that was a lot more cheaper and faster. Okay, and this was the start of the company Oxford nanopore device. And that is relatively so simple that this was used at the space station by this young lady who was who was at the space station, and they used it to do analysis in space. And this can be used in field, and this was used for the Zika viral analysis, and this was the Oxford nanopore method. And now I think that company has been bought out by I'm not sure by Illumina or other companies, So this has revolutionized, the speed and the ease of a DNA analysis in the field. Okay. Okay, yeah, the name of that, her name was Kate Rubins. Okay. Okay, and again, the sequencing cost has dropped further and further. Then they spent, almost, $3 billion for the initial analysis of the Human Genome Project, but the cost has exponentially come down faster than Moore's law, which, in transistor sequencing, as how the processing power has doubled every year, so to speak, in computers, the chip, as to how many transistors you can fit on a chip. But this has been even faster than the Moore's law in transistors. Okay, and this was from the Computer magazine from, I think, within last 2 months or so. The fast sequencing, so it's almost down to $200 for genome analysis. So you can see how much, dramatic changes that place. And the NIH's goal was to bring it down to $1,000, but now it has dropped down to $200 for human genome analysis. So this is, the amount of data that is being generated is exponential, and somebody has even gone down to $100 type of thing. So the cost of analyzing the whole genome, and as John mentioned just this morning to me, that at least in future, it may become that all newborns may actually start out with their whole human analysis. And whether one should do that or not, that remains to be seen, because we know how to collect the data, but we still don't know how to interpret all of it, type of thing. And again, with the massive data, it is becoming more and more easy to do huge amount of data analysis. For example, the Broad Institute, which is part of the MIT complex, they started a project to search for genes related to schizophrenia. Okay, and when they did about 10,000 people a genome, they still did not have much insight. So then they are amassing data of up to 150,000 people, to make sense out of creating what is known as a polygenic score. And the same thing is done for more common diseases as well, as we shall see, about that, to make sense out of the data and to create a meaningful signal, it takes larger and larger amount of data, type of thing, to make, reading sequences. Okay. Okay, so this is the cost analysis, and now at the end of the class, I think there are some companies that are offering this, at a really very reasonable cost, type of thing, but they don't give you the whole interpretation. So that's where you need somebody who's a genetic counselor to give an idea. And I shared my own data last time, so okay. And this is, now we already talked about how the DNA is such a long string of letters, and it's almost, like 2 meters or 6 feet long, but it's wrapped around varieties of protein molecules called histone.
In this animation, we'll see the remarkable way our DNA is tightly packed up to fit into the nucleus of every cell. The process starts with assembly of a nucleosome, which is formed when 8 separate histone protein subunits attach to the DNA molecule. The combined tight loop of DNA and protein is the nucleosome. Multiple nucleosomes are coiled together, and these then stack on top of each other. The end result is a fiber of packed nucleosomes known as chromatin. This fiber, which at this point is condensed to a thickness of 30 nanometers, is then looped and further packaged using other proteins which are not shown here. This remarkable multiple folding allows 6 feet of DNA to fit into the nucleus of each cell in our body, an object so small that 10,000 nuclei could fit on the tip of a needle. The end result is that the DNA is tightly packed into the familiar structures we can see through a microscope, chromosomes. It is important to realize that chromosomes are not always present. They form only when cells are dividing. At other times, as we can see here at the end of cell division, our DNA becomes less highly organized.
Okay, so that gives you an idea as to how this from, and this has a huge role. As this is how a gene that is how many thousands of kilobases away from another gene can manage to control the expressions, like the part, the intron part that is way down the road, so to speak, in the sequence, can affect the expression of that particular gene. So this is how the whole loop is come around, and this makes sense when we remember seeing the four-dimensional view of the genome type thing. So it's really, so it just qualifies my earlier statement. It's a mess down there, so to speak, so, but it works, so. And now this brings us to the idea that this was a book that's an older book, but it was one of the first book written by an English geneticist, I think this was probably, I should say about 10 years ago, Junk DNA. So that talked about these lesser known science of epigenetics at that time, and she's, so there are varieties of interviews of her that are available. The book is a little out of date now, okay, type of thing. So, and I just, this reminded me of the Junk DNA about this cartoon that was done by I think the dairy industry, that this was, I know I'm somebody, because God doesn't make junk. And now this is, again, the epigenetic factors, so let's just talk about, again, this is the video that you saw. Let's decipher it. And the chromosome is wrapped around this nucleosome, okay? And the varieties of factors, something called epi means above, and genetics means the genome that takes place. And there are varieties of mechanisms that takes place. And the first is called DNA methylation. That is a simple molecule, remember your high school chemistry, CH3 group that is attached to the DNA that controls the expression of this particular DNA element of the nucleotide. And then there are varieties of way that the histone, that is the little protein that is wrapped around, that is modified by another chemical that is attached to it. So, okay, called acetyl. So that also, and then last but not the least, is the RNA interference, that the small non-coding of microRNA, again, control the expression of this particular protein. So these are varieties of important factors that modify as to how whether the gene will be create the protein or not create the protein. And so these are the varieties of factors that control the expression of this particular gene. Okay. Now similarly, a lot of research that is done by a guy who has done work on PV or piRNA. Okay, and they showed that if this particular gene, the RNA, is suppressed inside the germline, then those particular cells will make less sperms. Okay, so this is, again, so it works at a very basic level as to how the RNA would control the expression of these particular genes that takes place. Okay, and they have shown experimentally in vitro that how if the piRNA is modified, then even the breast cancer cells will not migrate to a different zone. So some of these RNA molecules, which were considered not to be that important, and have a remarkable role in causing both health and disease. For example, like we give the example of the formation of sperms in the germline, and at the same time, that's one of the reasons why it may occur that two women may have the same BRCA gene, but one may get the disease while the other may not. So this is how the whole orchestra is played. Now, and now this was interesting that one of the persons who is, and I think one of my slides did not populate it, maybe on the other cassette type of thing. So what they did was they took varieties of rats and they exposed them to various environmental toxins, such as DDT or various chemicals, including pollutants and even hydrocarbons. And then they exposed the rat and they bred them to the second generation. And again, and even in the second generation, which was not exposed to the same particular toxins, okay, and there's one fungicide that was used in the wine industry and to prevent fungus infection to varieties of crops, and so that initially the rat line that was exposed to this particular chemical showed more incidence of tumors of varieties of organs such as prostate, testes, and it not only showed in the first generation, but the increased incidence of malignancy showed not only the second but the third generation of the rats that were bred, but were not directly exposed. Their genetic composition did not change, but the effect of the toxins or the environmental pollutants that they were exposed showed up even in the third generation. So similarly, some of the next third generation of rats became even more obese. So it just showed the dramatic influence of epigenetic modification that traveled along the not just the second or third generation. And the classic example that is given in the epigenetic literature was during Second World War, when the Dutch were living on starvation ration during the Nazi occupation of Netherlands. And they were living on like 800 calories. And even the grandchildren developed obesity because during when the food became available, people started overeating. And not only the second generation, but the third generation. So that is the classic example which is given, because obviously on humans, you can't do the same experiments and go intentionally expose people to a DDT and other toxins. But so this was what occurred, unfortunately, during the war era. So that is the classic example of epigenetic influence that is carried over the generations. Okay. So genetics versus epigenetics. So this is, basically the terra incognita, that was, we were thinking like the last large amount, which we classified as junk DNA, and a lot of them are, you can just see how many pi RNAs there are, 16 million, as opposed to the messenger RNA, there are 20,000, okay. And so this is a new world of genes, basically, okay. So pi RNA genes, they're hugely influential type of thing compared to traditional RNA. So there is a lot of work that is to be being done or to be done, so to speak. So this directs the epigenetic mechanism, such as stem cell renewal, and we already talked about the germline development. Okay, so this is, so it's a lot more complicated than when we realized, initially that it wasn't a straightforward A to C, but okay. And so these are, again, just to repeat myself, these are the varieties of elements of the pseudogenes that we talked about, which is different area, which has a lot to do with, like the vitamin C that we give example. Similarly, another interesting example was there was an enzyme which has to do with metabolizing uric acid, okay, which the other mammals have the element to metabolize uric acid, okay, but somewhere along the line, the chimps and the humans, lost that metabolism to the capacity to metabolize uric acid. And what's the price that we pay for it? We get gout, okay. Now you can ask yourself a question. I mean, why was that gene that is harmful not eliminated? And the possible explanation is that as with continued evolution, the change that took place, that climate became colder and food became harder. So it allowed this mechanism for people to store fat. So there was some beneficial, like the classic example we talked about when we talked about sickle cell of malaria, it gives some protection, one abnormal gene. So similarly, there was some advantage that was placed. And I'll give you the example of when fruit became more easily available, that's why the vitamin C thing was continued. So there's some interesting, evolutionary trade-off that takes place. Okay. And so these are, so how the interaction takes place between the varieties of gene and the non-coding RNAs to how it's a lot more complicated element that takes place. Okay. And this is, again, we can just see how the transgenic inheritance, that I already talked about. Let me see if I can, sorry. Okay, yeah. See how the next generation, the F1 was the first generation of animals, and the second generation, F2, third, and how the incremental continued increase of the tumors that took place. And at the same time, and the varieties of disease that showed up, and spermatogenesis, infertility, and so exposure to toxin wasn't as benign as you thought, type of thing. So it can, again, and just I did not put this slide just for sake of time, but along with this, somebody had done a research on something called environmental pollution and effect on cardiovascular disease. So it just showed that there was a direct correlation. This was some European data that I just listened to not too long ago, as to how even just the air pollution had a huge effect on the cardiovascular thing. So as to how that may modify, your gene expression to a considerable extent to be obvious in the data that is collected. Okay, and again, so these are the varieties of examples as to how common, even obesity and varieties of stress and so it's all sorts of things. So sins of your father can catch up with you, so to speak. So then, and the agents that were studied were the varieties of so-called carcinogen pesticides, dioxin, BPA, even jet fuel, and obviously, last but not the least, smoking. All of these had effect on the animals that were researched in this experiment. So again, and obviously, when you see the effect in the germline, then the information is passed on to the next generations. Okay. I mean, that was just okay. So, and this is how he showed the analysis of each and every chromosome. You can see how, and this is where the modification of methylation site that took place with this particular antifungal, meprozolin. And this is banned in Europe. It was used in wineries. Okay, I could not find exactly data if it is still used, but that use is being, I think, is being dramatically reduced now. It's only used for, I think in trying to think which one was it, and canola and maize. So it is still used as a fungicide. So these were individual genes. You can see the red arrow where the methylation, so defect can occur with this particular chemical. So you can see how the third generation showed wide varieties of exposure signature, or you can see that the fingerprint was left of all these elements that caused the changes. Okay, so it's mind-boggling, so to speak, as to how And this effect was not just individual, but there was, exposure was totally, individual. See how it all added up to the numbers. Okay. And so it was a huge fingertip, and there was no overlap, so if each one had its own effect type of thing of germline alteration. Okay. And this again, just to say how the technology had improved, this was there was interesting march from the Sanger method, where you could just analyze only a few bases to, thousands of bases to the nanopore sequencing, how the equipment has miniaturized to, the third generation this thing. So how it has become a lot more simpler to analyze, that thing. So, and again, I already emphasized the cost. Okay. And the Oxford nanopore, which was, which was sold out to some other company type of thing. So, okay. And this is not just, the DNA analysis that takes place, but there is also RNA analysis, proteasome, and my friend Dr. Johnny L. Ridey did a Friday lecture, I think, Friday lecture on Zoom last year called Omics, Omics and Oh My type of thing, and I think it was, and I think it's still available on Ollie website. And so, and he's one of a very well-respected teacher at USF. Okay, he's a mycologist by training. Okay. And so the varieties of omics that are available, are not just DNA, but RNA, proteomics. Okay, and then metabolics, where I showed the first slide earlier. Okay, so metabolome. Okay, right, and then also you can do something called lipidomics. And last but not the least, I mentioned something called exome. It's analyzing the pollutants that takes place and the effect that takes place. So we're awash in the amount of data that can be generated, and it takes a lot of computer power to analyze all this data and to make sense out of it. So we are awash in data, and as we have discussed earlier, it becomes a question as to who owns this data, as to whether it's your data or whether the company that analyzes this data. And obviously, a lot of you have read the book Henrietta Lacks, Cell Line. Okay, and it became a big, it was a very popular book written about the cell line that was used without her consent, type of thing, and it got very complicated. And so, and then there are varieties of use of genomics, type of thing. Just to give you some examples of it, the rare disease, which is where the genomic analysis really shines, prenatal genomic testing, and then and the other two big ones are pharmacogenomics and the cancer genomics. And ancestry is interesting, but in the end, that's kind of, we're all, if we go back far enough, we are all related to each other anyway. So that's, but that's a big popular testing, and we have learned. So we'll talk a little bit about each of the examples as we move right along. So we still have some time. Ancestry. So this is again, this is the brain size. You can see that from the earlier mammals, from the capybara, our brain size is increased dramatically, type of thing, And even though we have, we are a lot smaller than elephant, we have a lot more neurons, and this may be partly due to gene duplication. Okay, and this was done even before that type thing as to, and there were earlier geneticists. One was a New Zealander who was at UC San Diego named Wilson, and he and Mary King, who discovered the BRCA gene, they were doing some of the analysis of the genetic differences, at the comparative genomic analysis. They were the pioneer ones. And again, there are varieties of genes that have been of importance in terms of, the brain development as well as linguistic development. And a lot of them has to do with something called gene duplication, that the gene is copied over, from time to time. And some of the work that was done by a Japanese scientist who's not less well known, somebody called Susimo Ono. And so this process has been going on for a long time. And now, and as I may have mentioned, but just to repeat myself, that this might have to do with the availability of cooking and more available energy that became available as to, and just recently there was some data that the use of cooking may have started earlier than Homo sapiens and Neanderthals than we thought. So I didn't put a slide for that, but so there is some data which have benefited us to the human expansion of the brain size. And we have talked about how the chimp has to eat for almost a whole day to get enough calories for brain expansion. So it becomes like a mathematical impossibility to get enough calories for the gene duplication and expansion, so to speak. Okay, and the lady who had done a lot of the work by analyzing the nuclei inside the DNA, her name I have mentioned name, Susana Herculeo Hauser. She's a Brazilian evolutionary biologist who had done a lot of the science as to how the brain expansion may have taken place with the more availability of calories. Interesting work. And I think this, I think our colleague John Hassel has talked about it in his class at length about the human accelerated regions, that led to expansion and the difference between humans and chimps as to how there are certain areas that have expanded very quickly with the evolution of Neanderthal and other homogeneous species type of thing. And this work was done by California-based scientists, and they discovered something called the human accelerated regions that are only evident in human genome line, and some of the genes that are involved in it have a lot to do with neocortex expansion and synapse formation. So that and cortical development. So that may have contributed to the bigger brain capacity in human being. And the other part, there are about human accelerated regions in the genomes that were isolated. The other gene that shows on this side of the picture is something called the opposable thumb, which is called HARS2 region. And these are the example as to how the human and the chimp genome differs, as to this particular chromosome. So I think again, oh, sorry. Yeah, you can see how this is on certain chromosomes as to where the genes differ dramatically between the humans and chimp. And the numbers are academic, but it just gets the point across as to how the two big difference that we have with chimps. And here I have outlined in a nice table as to how humans have 46 chromosomes, chimps have 48, because two chromosomes are fused. Okay, and we have a lot more pseudogenes. Okay, and the endogenous viruses are different also. Okay, in the part, and again, some of this olfactory genes are different, and so there are clear-cut differences that are outlined, between the humans and genes. So even though we may be 99% similar, so these are the differences that have made us who we are. And obviously, one can see the advantage of the opposable thumbs. And I had talked at length about it when I talked about human evolution as to how the bipedalism had evolved and made a huge difference as well. Okay. But that gets us into the more human evolution testing. So I think so we'll move right along. And again, this is maybe of interest for classical ancestry thing out of Africa. And the earlier work that was done at the National Geographic as to how, with the comparative genome analysis that was done in various species, as we finally come to the conclusion that within the African group there was a lot more diversity that had given us the idea about the origin of Homo species to the African subcontinent, and how the migration took place across the land bridge into Arabia, into Europe, around the coastal line into South Asia, and across the land bridge into Australia, and across the bridge into the New World around, and that goes down to the earlier genome analysis that was done from something called from the area, which is the Clovis, which all of us, you may have studied in the high school history class. And that goes down to almost 13,000 years ago when the peopling of North America took place. Okay. And based on something called haplogroup, which are a group of alleles which are in an organism that are inherited from a single parent, and haplogroup, which is the same group, that is inherited in the similarities, we can decide the migration of the species. And this is, if you do your ancestry analysis, they will report your haplogroups. That thing. So, and just to throw in this interesting as to how it has been used, that as recently as the skeleton of King Richard III was exhumed from underneath a rock park in Lister, and based on analysis of his maternal DNA, it was conclusively proven that he had a rare mitochondrial haplogroup anomaly that was traced, that it was conclusively proven that was his skeleton. And his descendant on his maternal side was a woman who had migrated to Canada named Joy Ibsen, and her son had shared the mitochondrial DNA with Richard III. So how the genome analysis proved the ancestry tracing back to 1470, the current era. Okay, and this was the site where his skeleton was exhumed. And then, so this was the mitochondrial DNA, which is what you may remember, we talked about mitochondrial DNA, which is, which are relatively only 37 genes, but that are inherited from maternal side. Okay, and this is how they aligned. And there were some popular books that were, published, by an English geneticist named Brian Sykes, but this came out, I should say, probably around 2008 or 5 or something, called Seven Daughters of Eve, and that was a very popular book for geneticists. People said, okay, well, we are tracing back to the original Eve, but that actually talked about a group of people who migrated. So it was and this was for those of you who are Star Wars fans. Mitochondrial DNA makes an appearance in one of the Star Wars movies as well. And I did not know that, but this is, without the midichlorians, life could not exist, and we would not have the knowledge of the Force. They continued to speak to us, telling us the will of the Force, where you have to learn to quiet your mind and you will hear them speaking to you. That was the Anakin Walker. That was the younger Luke Skywalker. So it makes into the popular culture, the mitochondrial DNA. Okay. But that's one way to trace the female. And again, when I did my 23andMe this thing, it showed my haplogroup, which was, again, it's of general interest. I don't know if it means much. Okay. So this is, so hybridization has a huge, long history, not just for flowers. But this was an interesting report as to how, all of us know that Neanderthal and the newer species that was discovered, in the Russian cave, Denisovans. Okay, and this was a report from Nature that this was about how the mom was father was Neanderthal, mom was Denisovan. So there was a lot of hanky-panky going around at that time, So this was interesting. This genome analysis was done fairly recently, and this was an article in Nature magazine as to how. Okay, yeah, I'm sorry, I should go forward, not backward. And this was the Nobel Prize, which we referred to in the very first class, about the Svante Pabo, who did in 97, he did the mitochondrial DNA analysis for Neanderthal gene, and in 2010, they published the DNA analysis from a tiny fingertip bone, which was found in the Denisovan cave in Siberia. And so that was the discovery of the new species, And some of the gene is seen in the Tibetan population as well. So move right along. So where are you really from? So that's the question that I've been faced with many a times, where just to shut people up to avoid talking about football, I had to say Alloway, which is a tiny suburb of Green Bay, by the way. So it avoided all the discussion of Green Bay Packers when I was used to go to cardiology conferences. So diversity of race and disease, So again, we may say, okay, well, we're all very similar, but still there are some differences that are very apparent, and which is, like, and the classic example would be the Ashkenazi Jews, that some diseases are very common in that particular ethnic subgroup, type of thing. And there's some rough group, So, and this is the data that is done with the bigger analysis in New York City by the Mount Sinai group, because New York City has like this huge ethnic meltpot. And again, based on genome analysis, they were able to discover some of the newer diseases that occurred in the Puerto Rican migrants in New York City. And so there was some evidence of doing the big analysis. And again, the evidence of cancer was different also in some subgroups type of thing, as to see how average risk factor, okay. So some populations are how, you can see this was their data analysis that was done in various subgroups, prevalence of BRCA8 group. So you can see the numbers were, it's significant as to see 1 in 230 to, Southeast Asian 1 in 225. So the prevalence was, so we may say that, okay, well, and Mount Sinai is, they are starting a project of analyzing more than almost like 100,000 plus genes, they're doing, a lot of the big companies are doing this analysis. And again, similarly, the data analysis have said, when this was interesting that sometimes when the Europeans first went to Micronesia or Melanesia, they discovered a kid with blonde hair, okay, and they were just thought this must be some sailor who had, fathered the child type thing. But it turned out that there was a new mutation that had taken place spontaneously in a different subgroup, So this was not due to, travel by some European sailor, but the Melanesian blonde hair was caused by an amino acid change that had taken place. So this is again, so based on large data analysis, we're finding out a lot more than what we knew. Okay, let's see what I had. Okay, and this was just one mutation that had taken place. You can see that one amino acid change that had taken place. Okay. Okay. Now, again, getting down to the health part, which is what we really want to know. Okay, when to think about genetics, is to see how various, raise the flag, where there is family history. Okay, when there are a group of congenital abnormalities, when there is extreme present, like for example, totally unusual appearance, developmental delay, exceptional pathologies, and surprising lab values, type of thing. So this is when, obviously, because the whole genome analysis was still very expensive type of thing. So and the results that we get there, we get a lot more data, but it's hard to interpret. Like for example, clearly pathogenic, likely pathogenic, and this is the big elephant in the group when you do the whole genome analysis, because there is reported as variant of uncertain significance, and that becomes a question as to should it be reported or not reported, how to interpret it, because we don't have any data, and as to we don't know of significance in the present time, but it may become significant in the future. So doing genome analysis, when you're born, as to who would keep track of that data and how it should be reported, type of thing, that becomes a very ethical dilemma. And we still are ethicists, and the people who are doing the analysis are coming to terms with it. I gave the example about the HEAL analysis that's done for Mendelian diseases earlier, and some diseases are clearly so abnormal. So these are hard to interpret data. Okay, and there are varieties of hot areas that I already mentioned, as to how it has huge impact. Okay, and now where the genome analysis shines are diagnosing rare diseases. Okay, so the genetic diseases are a lot more common than you think. You realize they're about 1 in 5,000. So which may seem uncommon, but again, in the varieties of areas, the definition of rare diseases is different, because in US, because of larger population, 1 in 200,000. In Europe, the definition of a rare disease is 1 in 1,500. In Japan is 1 in 50,000. And there are varieties of databases and services that are available for rare diseases. The one, this is the North American Organization for Rare Diseases. This is a French website called OrphanNet for orphan diseases. Okay, so and this is where the gene analysis has really made a huge impact in diagnosing uncommon diseases. As to how, and these are quite serious, because they can have abnormalities of cognition, motor development, muscle development, growth. So these kids are really sick, and their prognosis is generally poor. Their treatment is not easily available. So that is where the genome analysis, and there are varieties of, and again, there are some human interest stories that have taken place. And this, I used to live in Wisconsin at that time. So this was a Milwaukee case about a kid who had huge problem with, his immunity, and kept getting repeated infection. Okay, and it was, impossible to treat. He had like almost 70 surgeries, before he was 5 or so. Okay, and then finally at that time again, so they requested, the team that was looking after him requested a whole genome analysis, and it was done, and it turned out he had rare enzymatic defect, and finally he received his sister's bone marrow transplant, and was eventually cured. And so that was, I think, about the 1 in a billion. And the reporter went on to write a book about him afterwards. And it was one of the earlier success stories of genome analysis making a difference in saving lives. Okay, and similarly example as to how this was another example of more recent, as to how the rapid sequencing made a difference in terms of how quickly the diagnosis was established, as to right after birth, as to how quickly the rapid sequencing, and they were able to find out that this child had a defect in the thiamine, various vitamin deficient metabolism. And this was, reported as recently as, 2021 in New England Journal. Okay, so it's a brave new word, and again, the genome analysis came into public knowledge when in 2013, the actress Angelina Jolie announced that she had a dramatic chance of getting breast and ovarian cancer, which was dramatically reduced. In the same way, genome research has shown this particular, and this is a slide from the famous George Church, okay, as to how one of his patients had the gene abnormalities, which made him more prone to getting blood clots and various eye defects. And so, and he was simple treatments such as taking antiplatelet effect makes a difference. Okay, and so this is, the variety of reasons how the genomic analysis as to why we get sick, and the your genetic factors, evolution, okay, at the same time, the other external factors that influence. So, okay, now again, this is a rather complicated slide, but I'll try to make some sense out of it. Okay, if I can zoom in without, okay, sorry about that. Okay, as to how varieties of diseases have effect, like for example, how much effect genetics has on some diseases, like such as autism, okay, or Alzheimer's disease, as to type 1 diabetes, eye color, as to, you can see how the eye color has dramatic effect of genetic. At the same time, some of the other diseases, far less role genetics play in disease, so I think it's, but I think I have to slide, but when you look at this individual slide, it would be easier to see as to how to zoom in on it, makes it okay, but it gives you an idea as to how much role genes play in certain diseases as opposed to more common diseases. As to, for example, in autism, the role is barely 50%. In eye color, it's 100%. Type 1 diabetes, a lot bigger role. Schizophrenia falls somewhere in between. Okay,
So-
and again, I'm sorry.
The celiac disease I saw on there on the left.
Yeah, it was very little. I think it was dramatically small than we thought type of thing. So it wasn't that, okay, see, it's not well clearly identified type thing. And as I answered your question last time, as a lot more defect on the microbiome rather than,
Genetic.
yeah, exactly. Yeah.
Okay.
And it may change in future, but at least right now it's still the jury's out, And again, and I may have mentioned when we talked about the medicine class, as to how treating people, instead of just treating blanketly for cholesterol, the LDL based on cholesterol lowering score and the polygenic score, you got more bang for your buck by treating. And this was the work that was done by the Mass General group, that was published in 2000 as to how the effectiveness of polygenic score, in terms of treatment, had impact. Okay, and similarly for Alzheimer's disease, the varieties of genes are people are still looking for sort of needle in the haystack. So we still haven't made a dramatic and a lot of the genes that seem to have effect on something called the cells that clean out the garbage in the brain, called microglia, and the inflammatory genes seem to have a role. And this is where the organoids seem to be making play. Some of the hereditary diseases, which we again talked about, in the medicine class, the classic disease, retinoblastoma, we talked about von Hippel-Lindau syndrome, familial melanoma, and some of the Lynch syndrome, or the familial polyposis, so these are the classical so-called hereditary diseases that takes place, and again, it's like when you, and the inherit, and this is how the genetic analysis is done as to how the people who are affected are the ones that are in dark circles, and how they, in the colon cancer patients, their children may have polyps, and then that is inherited in a dominant pattern if they have the APC gene abnormality, while the other ones who have sometimes de novo mutation that takes place, causing polyps, and there are some other diseases that are recessive. You can see that the probons may not be affected, but the next generation may show the disease. So again, the cancer genomics gets a lot more complicated, and but not the gene. The gene drive has been used for treatment for treating mosquitoes and Key West, and there have been genetically modified mosquitoes have been released in one of the islands in Key West. Okay, and that has made a huge difference in terms of Zika and dengue fever. Okay, and okay, anybody remembers which island in Key, in the Florida Keys was yet? No, we used to can speak up. Otherwise, I don't remember it offhand. Okay, okay, and so this was again, but there has been some community pushback as to see whether that's safe to release or not, because whether it will, but it seems like the overall benefit has been well established that it has. And this is how the gene drive works, because they're genetically modified mosquitoes, which only the males would be that, because the females are the ones that cause the disease, so that way the whole population is neutralized. Okay. All right, I think we're probably we'll jump over to couple of slides, and so varieties of the big pharma is interested in a big way in terms of getting the data, and Amgen had made this thing with the Icelandic project about the huge database that they had available, and they bought the data from the Icelandic firm Decode. Okay, and Regeneron is making contract with Geisinger testing. GlaxoSmithKline is collaborating with 23andMe. Okay, AstraZeneca is collaborating with Columbia, and so there's a huge work that is being done in terms of accessing data for the big pharma. Has a huge interest. Okay. And again, this is an old slide, but it's still worthwhile showing, because not all the drugs that we take are effective, and that is kind of, we may think that they're beneficial, but not all of them have the dramatic effect that we expected from, and some of them may have even more side effects. And it is much more common in terms of getting muscle aches from usually one of the most prescribed drugs cholesterol-lowering drugs, where you can get muscle aches if you have a certain enzyme deficiency in genes. And the same way, antiplatelet drugs, and the antidepressant drugs, some of these names, I can read, but I think you may have a harder time reading. Okay, Crestor, it's the cholesterol drug, Humira, which is used for Crohn's disease. Again, and these are the antidepressant drugs. And this is even gets worse for some of the chemotherapy drugs, that the response rate is very individualized and when we, and okay, and all right, and this was again, the drug that was of interest, I thought about how it was genetically synthesized, the Artemis, which was discovered from Chinese traditional medicine by a lady named Tu Yu, and she got the Nobel Prize for it. But it was harder to, cultivate, and so it was genetically synthesized from yeast by the collaboration from the Gates Foundation. But then again, what happened when the farmers in Asia started planting the trees, so again it became production cost it wasn't that much beneficial because they started overproducing from natural resources in China and Vietnam. Okay, but again, this drug was synthesized based on genomic synthesis. So where big pharma and other things came in collaboration. Okay, let's see what else I have. And again, genetics and the movies, which we talked about. Gattaca I had mentioned. Okay, the living proof was the treatment for chronic myelogenous leukemia. This was a movie done about the breast cancer, decoding the gene. I already talked about the 1 in a billion. So genetics has been in popular culture as well. Again, a couple of more slides, then we'll stop. I think so. I just okay. And there are some of the other companies that are offering personal genomic analysis. Another one which is financed by some of the big universities called Color Genomics, that gives you the analysis of cancer. And these are not horrendously expensive, about $200 or something like that. So they're doing this screening, whether it is worthwhile doing it or not. And this is the government program called All of Us Research, and you can still sign up for it. I think I signed up for it in the beginning, type of thing, and they're just and because of COVID, it was in the back burners, but they're beginning to release some of the data, type of thing. So this is and okay, so they are utilizing some of the information that you share for public and this thing. So okay, again, what we did not cover is genes and aging, genes and sex and reproduction. And last but not the least, which in 2008, George Bush II, passed the Genetic Information Act. So you cannot be discriminated against, for insurance and health benefits. So that was one of the great thing that was done. So again, but that again does not cover the long-term disability insurance and other things. But at least for health insurance, they cannot discriminate against you. So I think we are done. Sorry if we went for a few more minutes. And the last one is probably more garbage type of thing. So, okay. Again, if you own your data, basically, and this was the data from Nebula Genomics, which I shared with you last time. Okay, so I think we'll stop at this stage, and I will stop the screen share and stop the recording.